this webinar, which is going to be regarding compound management and actually using BioRails for requesting both inventory and managing plate inventory. The webinar will be conducted by our CEO President Andrew Lemon. And as housekeeping rules, I want to mention that you can obviously listen to this, but there's not an opportunity to interject as an attendee. Rather, what we ask is that you post your questions, and then we will answer them at the end of the presentation. There's a question area in your control dialog where you can ask questions and so forth. So without further ado, I'd like to have Andy get started. He has a presentation, then we'll go into a live demonstration, and then we will address questions. And if you have any difficulty with the audio or otherwise, please let us know through the chat box, and we'll actually try to remedy that. Thank you very much.
Thank you, Doug. So welcome, everyone. So today we're going to talk about sample management and review some of the challenges of modern environment towards sample management, and then show you some of our solutions in this area and how it fits together with the rest of the workflow tools that we provide. Before we start, I wanted to give a very quick overview of the company, and then we'll talk through some of the objectives of today's webinar and then go from there. So first of all, a little bit about the Edge. So we're about 10 years old, just over 10 years old. We specialize in being domain experts in the biopharmaceutical and pharmaceutical research areas, providing software tools to manage workflow and other aspects, data management for those sorts of companies. Our software is used by lots of companies. In fact, 4 of the top 10 pharmaceutical companies in the world use BioRails, our software, for various aspects of their research and development work. We have customers ranging from the west coast of the US right through North America into Europe and as far as Shanghai and Japan in the east. Our solutions are designed to deliver support for a number of different activities, all centered around the biologists. So we provide assay requesting tools, assay planning tools, as well as our core data management features in support of activities such as DMPK, ADME, in vivo sciences, in vitro sciences, screening, and biotherapeutics. We also provide the sample management software that we're going to be talking more about today, which is there to support the ordering and supply and tracking of inventory. If there's one thing we're passionate about, it's helping you to optimize your processes and understand the process that you're using during your research and development work, and making sure you keep the value in your data. So this is why we founded the company on the premise of keeping structured data and maximizing its use. So the objective of today's webinar is to review our sample management offering, look at some of the challenges involved in a modern drug discovery and development environment and supply of samples. We'll talk a little bit about our solutions, and then we're going to see it in action with a demonstration that covers the full workflow. Finally, we'll review the benefits and then take any questions at the end. So a little bit about some of the challenges of a modern environment. So obviously there's been a great deal of changes that have gone on in the last sort of 5 to 10 years in this pharmaceutical and biopharmaceutical environment. There's a move much more towards more networked research and development models, working with partners, contract research organizations, and also academic partners. So we need a software that supports that sort of environment. Also new business models. You know, everything is not always done in-house these days. There's a vast array of different options there between working with external organizations, insourcing, and as well as running things in-house. We also have to manage the supply chain or supply workflow within the overall workflow of the organization. So we're going to drill into some of these 3 challenges here and talk about the specialities of the challenges in those different environments. The first one's around networked R&D models. So this means Working with CROs, partners, collaborators, and other organizations means we've got to cross different boundaries. So there are obviously geographic boundaries. We might have a contract research organization that's synthesizing compounds or generating biologics for us over in China or in India, whilst we're doing our biological testing perhaps in-house in North America or Europe. Or we might be using contract research organizations for doing, or academic partners for doing some of the testing work at various other locations in the world. So obviously we're covering geographic boundaries, also organizational boundaries. So we need to take into account things like security, who's allowed to access what, and build those support into the software that meets these challenges. And of course, classical disciplinary boundaries. So the interplay between project teams generating new entities and test teams that are testing them and other people that are involved in that workflow. Second area, these new business models. So we need software that can accommodate processes, compound management and sample management processes that are run in-house, or they might be outsourced to contract research organizations, or indeed they might be hybrid models where we're running some things in-house and other things out with the CROs. Today we're going to show you an example of a deployment of BioRails, which covers this hybrid model. You can use BioRails either to support in-house processes or support outsourced processes, or indeed to support a hybrid between the two. And today we're going to be talking about an example of using it in a hybrid environment. In this particular environment, we're using an off-site powder shore that's located out with a contract research organization where vials of powders are managed, and then they're shipped and dispatched between the host organization and the CRO, and then back and forth. So we've got to manage the sample dispatch process, receipt process, and also make sure we have a real-time view of the inventory. So that means making sure we're kept up to date on any inventory changes, both at the contract research organization and also internally as we use the materials. In this particular environment also, we're using on-site liquid stores. So we've got highly automated systems that are managing plate preparation, storage of liquid tubes, and of course transferring and stamping plates and racks of materials so they can be used for assay groups and preparation of assay-ready plates. The third area of challenge is around workflow. So obviously there's a lot of workflow getting involved in an organization, both receiving inventory orders from assay groups that need them for their assays, or other scientists such as formulation scientists needing powder to be ordered so they can formulate the compounds. We need to make sure we're sourcing the tubes from the right location, so to optimize the turnaround time for assays, for example, and then make sure that we can prepare assay-ready plates and vials ready for laboratories. And that's true whether we're doing the plate prep within a sample management facility or whether we're doing it in the assay labs with, and just ordering up the tubes or vials even. And here's an example lab workflow. So it starts off with a project team that are generating demand for assays. So they're selecting the best lot, that's a unit of manufacture, of their particular material they're testing. So it could be a biologics company, operating on proteins, antibodies, or conjugates. They need to select the materials they wanted to have tested and then submit into a set of assays that are going to be run. And then for the assay teams themselves, they're going to be reviewing the workload that's coming in, the demand, and then obviously sourcing and evaluating the inventory that's available for that. So in this particular example, we might be looking for liquid tubes that are in a liquid store like a Hamilton tech cell instrument. So there, obviously, if there are enough tubes in the tech cell, I can immediately move forward to ordering and preparing the plate. If there aren't enough tubes in the tech cell, then I'm going to have to go away and source the powder information tubes so that they can be transformed into liquid and used for my plate preparation process. So again, here we're interacting between project teams, between assay scientists, and also between the compound logistics or sample management groups to manage different aspects of this workflow. So we need a system that's going to support us during the workflow to optimize turnaround and make sure everybody's kept in touch with the workflow that's going on. So we've been BioRails has been around as a product for about 9 years now. Version 1 was right back in 2006. We originally developed as a data management platform and have evolved that to support more and more of the workflow centered around the biologists. And today we're going to be focusing in on our inventory module, which is used to support the processes we just mentioned. So it's a web-based inventory software built on the BioRails platform. It handles assay requesting as an option to drive demand, or indeed you can use it together with already existing assay request tools. It manages the workload so you can distribute it across the scientists that are running the assays, and also helps them to support the plate preparation operations. So that's ordering inventory, delivery and transformation of that inventory into assay-ready plates, and of course being able to track quantities and locations of materials across the organization or indeed across organizational boundaries. In the example here, we've integrated with an off-site powder store through a set of web services. So this allows us to manage transfer of inventory between the offshore powder store and the local storage, and also all of the plate operations that are used to prepare plates, allowing you to track and update the workflow. So we drill down on this workflow a little further. It all starts with tracking the demand. So here we have an assay requesting module, which is used to track assay requests and also automate the generation of orders for inventory. So this means basically as materials are generated, synthesized or generated and registered in the system, we can trigger orders and requests for the assays that are needed to be required to be generated. We can then track and receive those processed orders. We're keeping all the scientists involved during the workflow. We can also automate the transfer of materials from one site to another in order to minimize turnaround times. So that's anticipating that material that was synthesized on one site is needed on another and needs to be transferred. There's also an ordering interface to allow you to evaluate the availability of materials, identify source containers from across different sites, and then issue orders for those source containers to be delivered to the right location or person. There's an approval workflow built into the software. So if you've got restrictions on compounds and you need to have approval by project team members in order to release the materials, then that's all managed as well, including email notifications for various aspects of the workflow. And of course, once we have our the right source materials available into compound management, we also support the process of plate preparation. So this includes dissolution of powder samples into liquid tubes and placing the liquid storage devices, and then creation of racks of tubes that can be then transferred into assay plates, including stamp operations, quad mappings, dilution series, etc., and control of controls and standards as well. And of course, we'll need in that support for some automation of some of those processes, such as driving liquid robots, etc. The final thing now is when we've prepared the plates, we need to track their delivery to the assay teams. So that includes dispatch and receipt of containers from remote inventory stores, tracking assay-ready plate orders, and then making the plate maps and assay details available to the assay scientists so they're ready to run their assays on their equipment. And of course, the BioRails platform also continues to support them for scheduling their experimentation, automating the reading of files from their reader instruments and processing those into results, writing up their experiments and signing off the intellectual property. So we really are tracking the full workflow. Here's an example of 3 customers that are using our software at various aspects of that workflow. So the first one is Zoetis Animal Health, and they're using the full workflow support for animal health discovery. So everything from assay requesting and planning activities through ordering of samples, and then the sample preparation and inventory support right through the testing process and then storage of results, all within the BioRails platform. Another customer is AbbVie. So they're obviously a global biopharmaceutical and pharmaceutical company, and they use a highly automated large-scale deployment of BioRails for managing assay requesting process, coordinating the inventory ordering, and assay scheduling as well. And we've been working with them since around 2009, integrating our systems with their various logistical systems to manage the workflow. The third example customer is Evotec. They are obviously a contract research organization offering up both assay services and also the compound management services as well. So they're using our platform for supply of the inventory supply for DMPK testing services, obviously integrated with local powder store facilities as well. And of course, there are many more customers that we could talk about. So what I'm going to do now is I'm going to give you a demonstration of the system, and we're going to run through the workflow. The example I'm going to give you is very much going to be a sort of manual running of the workflow, but there are many opportunities in there to automate the steps, either directly or through integration with robotic systems, etc. So these are the three characters that we're going to be talking about today. There's Walter Skinner. Walter is a project team leader, so he would run a biopharmaceutical project or potentially a small molecule project, and it'd be submitting lots of material to be tested across various assays for the project. Barbara Bryson is then an assay scientist, so she works in one of the assay labs. In this example, she's working in a DMPK lab, evaluating a DMPK profile for new entities that have been generated from the project team. So she's going to receive that request for those assays, going to be evaluating the inventory, and then ordering the inventory to be prepared from the sample manager. And then John Doggett, he's a sample manager, works in the sample management group. He's going to be receiving powder samples from the offshore powder store and then processing those into assay-ready plates, ready for delivery to the assay lab. So this workflow involves 3 different people. So hopefully you can follow it as I run through the demonstration. Just try and keep track of the fact that I'm changing the roles as I go through. So we're going to start here. So this is BioRails. It's a web-based application, so I'm just using a normal web browser to access it. So the first thing I'm going to do is log on as Walter Skinner. And Walter, as I mentioned, is a project leader. So Walter can search the inventory to check stock levels for his compounds that he's going to submit. He can monitor the full project workflow using something called a cascade, which includes managing progression decisions between tiers of different assays. It can also generate individual requests. So I'm not going to run through all the requesting process today because that's the subject of other webinars, but I'll just show you this first request here. So this was a request I made earlier on for 8 of these compounds to be tested and evaluated in 2 assays. So 7 of the compounds are going to be requested in this microsome stability assay, and then all 8 compounds are going to be evaluated for liability for a CYP P450. Actually, it's a panel of 3 different CYPs, so it's going to require 3 different assay plates. They're dose response series to be evaluated to calculate the IC50s for that. And that's been requested by Walter and requested into the system. So that's all I'm going to talk about for Walter. So I'm going to change hats again now and log in as Barbara Bryson. So Barbara works, she's a lab manager in the DMPK group. She actually runs 3 assays herself, this P450 IC50 inhibition assay, plasma protein binding assay, microsome stability, and also a PK in vivo PK assay. If she wants to view all the work that's going on in her group, she just clicks on the View Assay Queues, and it will show you all her team's assays. So immediately she can review maybe requests for bioanalytical results of tissue samples. We've got the assays we just described, things like blood plasma ratio, PAMPA, and you can see in here, you can see the number of pending requests items, that's samples to test, and also the number are being worked on. So in this case, we're going to go through and have a look at this P450 IC50 inhibition assay. So we'll click on that and it'll show me the detail of the samples that have been submitted. So here there are a number of lots that have been submitted to this particular assay. I can see they've all come from Walter, from the chemistry team, and they're made on behalf of a project, or in this case it's a customer because I've set this up like a contract research organization, but you can tag it with project details that can come from your databases and your list of projects, or in this case it's a list of customers, so I'm just saying customer A. So those are the request items. If I scroll to the right hand side here, you can see that we're requesting all these assays against a panel of 3 SIPs. So I've got SIP 1A2, SIP 2B6 and SIP 3A4, and I could have other SIPs, and here I'm asking them all to be tested against a particular concentration value. So what I'm going to do now is Barbara wants to evaluate basically whether there's enough inventory available for these samples. So what we're going to do is select the samples, we put them into a cart, and in the cart what we're going to do is we're going to evaluate to see if there is enough source tubes from a liquid store to meet the needs of the assay. And you can see immediately that 7 of the samples have tubes available with enough material to test in the correct location. 1 of them doesn't. So how does the system know how to evaluate those? So it does that because it has something called an order profile that comes with the assay. So here's a link to the assay queue itself, and I can see here there's a link to the order profile. So in this order profile here, we can specify basically both the source where the tube should be sourced from, which is specified by something called a requestable collection, which identifies a subset of samples that in this case are in a Hamilton tech cell instrument. Could be other locations and other instrument stores. Also, we're specifying the amount that we want from each tube. So we're looking for 30 microliters of 10 millimolar stock, 10 micromolar stock in this case. And we want them made up in 100% DMSO. In a plate, which is a 96 well, 8 compounds down, 10 dilutions across, no repeats, and then I want 100% inhibition and a 0 inhibition in the 2 control positions. I want 3 of these plates because I'm running a panel of 3 SIPs, and if you want to, you can review the plate format itself, where all the materials are. So it's a high control, that's the low control. And even if you want to, you can specify what controls and standards you want to put into that plate prep position so that a complete plate is prepared and shipped to the lab. In this case, I'm going to leave out the controls and standards, and the actual assay lab themselves are going to put those on the plate. You'll also notice on here, if I want to, I can vary the volume and concentration per well of the plate. So I can fully define what the plate is itself. I could have different solvents as well, and concentrations and other elements. And those can actually come, so each sample could be different volume, different concentration, etc. And here I'm also specifying the dilution factor, so I know between each dilution in the series, I can tell basically how much is being diluted by. So that essentially provides you with all the information for a templated order from the ordering system. So that means from the assay queue itself, I can issue an order for those containers. Now, at the moment, I know that there's only enough tubes in here for 7 of the compounds, and I want a complete plate. So the first thing we're going to do now is we're going to order some powdered material from the powder store to be solubilized, ready for the tech cell instrument. So to do that, go back to our home page here, and I'm going to run a search against the inventory. So I'll just pick up the sample number that's required, or I could export them out to a file if I wanted to, and then I'm going to run a search on the inventory. So here, this search page, you can do structure searching, if you like, that's a criteria. In this case, I'm going to specify I know which lot I want. And what I'm looking for are bottles that are in the powder store with the CRO that have about, say, 1.8 milligrams of material available in them. So let's have a search and see if I can find any of those. So here we go, there's one here. That's the tube that's available at Evotec, at the powder store itself. So you can see here where it's located. And you can see there's 8.2 milligrams of powder available at that powder store. You'll notice there's also already an order for 1.8 milligrams that's reserved. There's actually 10 milligrams in the current container. So you can see we can have multiple reservations and track those as well. So what we're going to ask for then, we're going to order for that material to come out of this container and then be shipped in a new container and delivered to the laboratory. So we'll do that by putting that container in the cart. We're going to proceed to the order. Select the compound. I'd like it in a matrix tube. Okay. And what I want is 1.8 milligrams. Oops. If I want to, I can also specify volume. I can order the entire container, or I can order a molar amount as well, and it will calculate the amount as well. So I want that delivered, in this case, to the compound management group and into the storeroom and freezer 2. And I want to do that on behalf of a particular project, let's say customer A in this case. Again, that could be a list of internal projects. If you're an internal team, that's fully customizable as well. And I want just one sample because that'll be enough. And I'm just going to say for a SIP assay panel, just to let them know that. And you notice here, well, as I filled in the amounts, the availability was evaluated. So it's saying there's an amount available, however, it's pending approval because this particular sample has a protection on it. So let's go through and place that order. It tells me here, here's my order number. And you can see I'm asking for material to be taken from this container, 1.8 milligrams. That will leave the available amount 6.4. I want it shipped to this location on behalf of this project, and it's for this SIP panel. And you can see it's awaiting approval by Walter Skinner. So before this order can proceed, Walter's going to have to approve this. Now there's an automated email approval request that's sent to Walter with a link to it to click to. So I'll show you where that goes. So I log in as Walter. There we go. And it will go through essentially to the order items that are requiring his approval. So these are pending orders, approval by me, and I'm now Walter Skinner. So if the email link will take you straight to this position here, so I can then approve the order. And I might have multiple orders come from lots of different assay labs that are all in here waiting to be approved. So we'll just select that individual order item, pop that on the list here, and then I can either approve it or reject it with a reason. So I'm going to approve this one. So that's my approval done. So now I can log in as, I'm going to log in as John Doggett now. So what we're going to do now is we're going to skip through that workflow. That order will reach the contract research organization. They're going to measure out 1.8 milligrams of powder into a container and then ship it to the compound management group. Compound management group can then will receive that order, and there are features in the product for doing dispatch and receipt of containers that you can do from in here, from the administration area. In fact, it's coming from the tubes and bottles query in this case. So you can see on here there's support for dispatching materials and also receiving materials from the CRO. So what we're going to do, in fact, is to pick up this example here that I've already prepared. So this sample has been shipped to us and received through the receiving process. and here's my sample. What I want to do basically is to solubilize this powder sample and then prepare it and put it into the Texel instrument ready for the assay. So to do that, I can put multiple tubes, powder tubes into a rack and then solubilize the rack, or I can solubilize the individual tubes. So I'm going to just do the individual tube here. And so here's my tube. It tells me basically how much material is in it. So here you can see the level that's required. I've got the reserved amount and the actual amount that's in it. And I can come in here and just press the dissolution button. I'm going to be putting these, I want to dissolve these into a 1.4 milliliter matrix tube. And I'm looking for a target concentration of 10, in this case, micromolar. So that will tell me, oh, sorry, millimolar. So if it's micromolar, then that's a lot to dissolve it by. So we'll just make it a millimolar one. So it's telling me to add that much solvent to this liquid to get it to dissolve. So then what I do is record the actual amount that I've added. So let's say I've added 0.95, whoops, let's say 96 milliliters of liquid, oops, milliliters. And I'm making it up I'm adding DMSO at 100%, or it could be another combination of solvent. So let's dissolve that sample. Here we have it. You can see it's a quite full tube now, and I've got, this is the concentration. It's nearly 10 millimolar stock that's in there. So if we go back now to our assay queue, so I'm going to log out as John Doggett. Oh, the last thing I need to do is then place this tube into the Texel itself. So to do that, we're going to put it into the requestable collection for the Texel and also locate it into the Texel location. So here it is, it's Texel, and I'm going to put the tube in this location and save that. And then now that tube is in the Texel instrument ready for the assay. So if I again log out of here now and log back in as Barbara again, do apologize for having to keep swapping, but you can see it's a workflow that involves more than one person. So now if we go back to our queue here, which in fact is shown here, and reevaluate the stock levels in it, you can see it's now got a tube for every sample. Every lot that's been requested has a tube available in the Texel, so I can go ahead and then order those containers. So all I need to do is click the order button. That will gather up the information from the order profile, which I'll remind you of here. Here we go, let's go to the order profile. So it's going to basically ask for Texel tubes with a certain amount in it to be made up into this format and shipped to this location, which is the UK ADME DMPK group in the ADME store 2 into shelf 1. Okay, so and that's the request that it's made. It's submitted this set of tubes for an order to be made up into the assay ready plate. So as the assay scientist now, I've issued my order. I just need to track its progress. Once the order is complete, I'll receive an email with details of which plates are going to be shipped to me, and so that I can get on, pull down the plate maps for those plates and get ready for the assay to itself. So I'm going to go back now to John Doggett. And John's going to monitor his requests. So here we've got the orders coming through in this matrix tube to plate service that's available here. And you can see these 8 tubes now have been received in an order, which is for the SIP IC50 assay. And it's asking me to make up this plate layout from the requestable collection Texel, so I know which instrument to get them from, which store to take them out of. It also tells me details of the order parameter, the top concentration value, the solvent, etc. in the order. So you can see it's DMSO solvent at 100%. Here's the individual volumes. These could all vary. Depending on the assay, you may have variable amounts for each one. So what I'm going to do now is I'm going to process this order. So what I'm going to do is basically select the 8 tubes. I can then grab the tube names. So here's my tube names. I'm going to take those tubes out of the text cell then and place them into, so let's, I'll open that on a new window. I'm going to take them out of the text cell and put them into a rack. So I'm going to create a new rack containing text cell tubes, and I'm going to use this 96 layout matrix tube rack. So I just paste in the list of tubes and add them. And then I want that rack to be barcoded, so I'm going to pick a barcode for the rack. So I use this HTS barcode generator, and that will automatically generate the next barcode off the sequence. The barcode generator can be administered. You can choose the formats, the prefix, and the sequence number, and the system will automatically generate barcodes. So we'll click next. Okay, and then you can see it's created this new plate with a KHTS prefix and a sequence number, and it's got 8 samples on the plate for the 8 compounds that I've just, the 8 tubes that I've taken out of the text cell and put into the rack. So all we need to do now is place that into a local storage. So we'll put that into the storeroom freezer, into that position. Click next, and just confirm that now. And then that will generate that new rack. So here's the rack it's created. So we just open that. You'll be able to see a graphic for the rack. You can see there's my samples. I can look at the individual samples and see which lots are in them down this rack here. So what I want to do now is take that rack and create a dose response plate from it. So to do that, what I'm going to do is transfer liquid from the column and then stamp it out into a dose response series. To do that plate operation, I'm going to use something called a plate transfer. So again, we'll use the wizard. We're going to use a plate transfer. And the plate transfer is based on a particular style. So the first thing we're going to do is it's going to be a store to test operation. And we're going to take text cell tubes, and then I'm going to use this transfer map. So what the transfer map is, it takes a 96 position matrix tube rack and it stamps it out into a 96 well dose response plate. So it includes taking multiple liquid transfer operations from the source tubes and then out into the resulting dose response series across the plate. And I created this as something called a custom plate map. There are wizard support for defining plate transfer maps for both dilution series, stamps, quad mappings, all sorts of different transfer operations are available. Next, what I want to do is then select the source plates I want to operate from. So here I'll just select the plate that we just created, add that to the list. So that's my rack, sorry, that's my source rack in this 96 position matrix tube rack. And I'm going to create 3 replicate plates because that's what the request was for, with a top concentration of 10 millimolar and then a final volume of 10 microliters, dilution factor of 1 log unit, and in 100% DMSO I'm going to use a Hamilton star robot, and I'm going to generate a plate with a barcode, an ADME barcode. Okay, so obviously this will record the transfer operation, and then you can actually use the automation software with the Hamilton star robot to process the final order to actually generate the physical plate. So we'll do that, and that's going to stamp out that plate operation to create the virtual plate before I go ahead and create the real one using the robotics equipment. So it's generating the plate. As it's doing it, it's checking that there's enough material in the source tubes to match. So you can see here that one of the tubes does not have enough material. That's because this should have been micromolar, not millimolar. So let's, in fact, I think I wanted 30 microliters as well. So I got that completely wrong. Okay, so let's try that operation again. Got to pick the barcode generator. So it's now preparing that plate. And again, doing the same stock check. So hopefully this one, there's enough materials available. So it does a number of checks on here. It does, it checks to see if there's enough moles of material in the source tubes to make up the final plate. It also checks to make sure that the volume of the final well doesn't exceed the maximum volume of the container. So here from that one source matrix tube rack with the 8 samples on it, I've generated 3 plates, which are HTS plates for ADME. Each has got 80 samples because remember I'm generating a 10-point dose response for the 8 compounds and then replicating it 3 times. So all I want to do now is then place that into the destination location, which is UK, ADME stores, ADME store 2, and we wanted shelf 1, and I'll put it in position B in this case. So all 3 plates can go in there. All of these operations can also be automated, either using files-based mechanisms or also using the web services as well. Okay, so that's just summarizing the final details of the transfer. So here's my source plate, taking 8 samples from the source plate and then creating 3 plates with 80 samples each on to be placed in this location. So I just confirm that now, and there is my my plates prepped. It'll take me back to a list of the plates. So here you can see here's the 3 plates. If I want to have a look at the details of one of them, I can do that. But this is the transfer that it's recorded. You can go back over the transfers. It's decremented the source plate, so it knows that I've taken material out of this rack and out of the tubes in the rack, and I've used them to create these destination plates. And here's an example of the plate. So you can see I've got nothing in the control positions at the moment. If we go across the plate here, you can see here's the different samples. I can see which lots are in them, and then I can see the dilution series that's going across the plate. And it gives you a graphic just to show you the dilution. From this destination plate, I can download the plate maps. Once I've completed the order, then Barbara Bryson will receive an email with a list of the plates that have been generated for her order, and she can, from the link on the plate, she can quickly go to the plates here and download the exported plate maps from the list of plates here as well. So that's that part of the order completed. So I'm now going to go back in as Barbara. So we'll just log in as Barbara. Sorry, it's B. Bryson. So if we go back to the assay queue again, you'll see now these items are now in an ordered state. And again, once she receives the email with the details of the order, then she can select those now, download the plate maps, select the samples, and then go ahead and schedule the actual experiment itself, which is called a task. So we'll do that now. And here what we're doing is we're creating an experiment, and we're going to run a particular workflow. That workflow includes information about how to test the protocol for testing this particular SIP assay against the panel of SIPs. It also has a data analytics template attached to it, which will allow us to process the reader files from the instruments for the SIP assay and process them into results that can be posted, signed off, and then results can be dispatched back to the project team. She can also assign the experiment to other members of her team, as well as giving schedule information. I'm not going to go into this because it's not part of the today's webinar. If you want to see details of that, you can go back and look at some of the previous webinars, and there'll be future ones talking about the assay requesting experiment management processes. Okay, so just in summary then, go back to the slides. What I showed you was how Water Scanner generated a request for a project. That request was a particular assay that was run by Barbara Bryson. Barbara was able to evaluate the stock levels of the source material required to make her assay-ready plate. She identified the fact there wasn't enough tubes in the Texel instrument, and then was able to order powder to be shipped to sample management to be solubilized and placed in the Texel, and then she could submit her order for assay-ready plate again to the sample management group, so it could be processed from liquid tubes into a rack, and then finally into a fully defined assay-ready plate, shipped to Barbara, where she can place her controls and standards on it, schedule the experiment, and then run the assay itself, delivering results which can be analyzed using other parts of BioRails, and then results published back to the project team. So we've got this full workflow here. Hopefully that was reasonable to follow. I know I was jumping around a little bit, but hopefully you managed to follow the workflow. I showed you a very much manual workflow, but there are lots of opportunities to automate that workflow. So that concludes the demonstration part. I'm just going to quickly review the benefits, talk a little bit about some of the advantages and future directions, and then we'll take questions at the end. If you have got any questions, please do fire them into the questions panel, and we'll go through them. Okay, so a little bit just to round up some of the benefits, what I just showed you. First of all, this inventory system is absolutely not fixed on small molecules. It works with any type of inventory material. So you can use it for tissue samples, you can use it for biologics, small molecule compounds, samples. You can use it for pretty much anything. It just takes a particular sample type that you define. It will track its quantity, location, and if it's a liquid sample, allow you to do liquid operations on it. It's a very powerful system that's easy to configure for your particular workflow, so it makes it accessible for more users. It contains a flexible plate format, so it can be used to define plate formats for a whole range of different assay types. So both plate-based assays, obviously for the plate formats. If you're using non-plate-based assays, you can define racks for testing from. You can also define and have assays that are based on powder order profiles, so they're ordering powder sample rather than liquid. So things like formulation services, it might be in vivo tests where I'm defining the amount of powder I'm going to formulate for dosing. It's all integrated within the overall assay workflow, as you've seen. So it helps improve communication between project teams, assay teams, and assay teams and the sample management groups. Helps to smooth out the workflow, improve turnaround times, and automate much of the steps that are traditionally manually done. So it basically means once you're ready with your step of a workflow, the next person will be informed and can pick it up from there. For IT, it's a fully web-based system, so that minimizes deployment effort, means you can make both local and remote users have access to it. It's also got an extensive set of web services for ease of integration and also automation as well, So some of the advantages of this system and unique aspects is, first of all, it's this overall project workflow and just being able to connect all these people together. We also have support for cascades with automated progression in there, so you can automate decisions. It's all workflow driven and fits together with the data management system. So one part of the workflow fits together with the other and helps to drive matters through. And it manages all aspects of the workflow, as I said, from project teams through stock checking, ordering process, through plate prep and delivery, through experiment execution, into data analysis, and even into progression. The platform itself also facilitates data exchange with contract research organizations and partners as well. So you can pass experiments off to third parties. There's templating support, so you can email templates to be completed and receive data through email, or you can allow them into the system to actually load data directly through the system. So many aspects for allowing a collaborative network environment. Our future plans, we're working on a brand new platform user interface to simplify usability. There's more order tracking support in the next version, more plate transformations, and we're also providing an extensive set of web services to support the automation processes as well, over and above the ones that are already there. What this means is you can actually put your sample logistics as part of your overall workflow. So rather than having to communicate via email or files, you can actually use an integrated system across the whole platform. So at that point, I'm going to hand back to Doug, and he's going to take us through any questions you might have.
Thank you very much, Andrew. We have several questions, so let me start with one. How can the lot or best lot decisions be customized?
Yes, so that's often a question. So what we do there, again, it's a very modular product. So basically, we've implemented a standard best lot workflow, which goes along the lines of evaluating based on location, based on quantity and format. So, for example, in the example assay I just showed you, obviously it was looking originally for liquid samples in the tech cell in the location of the assay. If those are unavailable, then it goes by default, it will look for alternative format on the location of the assay. And if they're not available, then look for the assay format available at a different location. That, however, is all encapsulated into a procedure which we can modify to your particular business logic. So, because we obviously realize different organizations have different setups. The other alternative is that you evaluate the best lot independently and then just supply the lots. So it depends whether you're requesting based on a concept, a compound, or a lot. So hopefully that answers the question.
Good. Next question. Are there any limitations on plate size or format?
So we have quite a powerful set of plate layout definitions, which allow you to do things like repeating series, repeats either by dose or route in the direction of repeat. So we think we've got a pretty extensive set of plate layout options for handling different plate formats. In terms of size, we've customers using it for the full gamut, right from 96 wells right up to 1536 assays. So and we're working also on optimizing the support for those as well. You can actually test any size plate you want. You just define it in terms of its dimensions and then its layout. So it's a very flexible system.
Thank you. Next question, can the order profile details be customized?
Yes, yep. So there's a number of ways that can be done. We have a standard order profile, and then if necessary, we can create custom order profiles if you need extra information on there or more details on it. But you saw the standard one there. It includes support for dilution series and everything else there too.
Good. Next question, can we specify an in concentration rather than dilution factor?
So in the current version, it's based on a starting concentration and dilution factor, although that's an interesting thing. I don't see, in principle, we could do it the reverse direction as well. So start with the most dilute and then, if you like, concentrate it back with the dilution factor. Or indeed, you could specify the exact dilution of each well as well. You can do that as well.
And then does the volume always come from the order profile?
No. So that's the most simple case. So if I just swap through here, we'll go and have a look at the actual assay queue itself. So let's go back here. And we'll look at our assay queue. look at the order profile here. So you can see in here, I've specified the volume, concentration, and dilution factor here. If I want to, I can put them in hardwired. They can also come from the process itself. What that means is individual request items can have customized versions of the volume. So you can actually specify in the assay queue itself which volume and vary it per sample in the thing. So you can actually, create a completely customized plate format from the assay queue itself. So the order profile standardized, all you do is you just pick. So here we can see if we look at something like the concentration, top concentration, I could pick that from the order profile itself. So actually it's not always 10 micromolar, it could be customized for every sample. Okay.
And then the next question, can the inventory system track the same lot in two different locations, i.e. solid at a CRO and solid in-house from the same synthesis?
Yep, so the lot is, of course, the lot is still a conceptual thing. It's a lot of manufacture. So, and it's realized as a number of different samples that are in containers. So you can have the same lot in multiple containers that can be in different locations. I think that's the question.
Yes, whether it can essentially track the same, essentially, lot and batch at different locations.
Yep, you can certainly do that. And of course, each one of the individual samples themselves has audit trails on it, so you can see. So if we look at something like tubes and bottles here, let's look at one I've shown earlier on here, this one. I can view the history here as well. So it shows me when it was created, how much was in it. And then you can see as we've used the process, we've been decrementing the amounts in the plate. So this particular tube started off as a powder tube, and then it was transformed into a liquid tube, and then material's been removed in order to make up a plate in the particular format using a plate transfer. So there's a full audit trail on who and when and what.
Good. The next question, is there a plate designer?
Yes, so if I go into the administration section here, you can see here's the BioRails inventory module. And there are a number of different features in here. So you can manage and you can generate barcodes. So here, if I click on the link here, select a barcode generator, which you can set up inside the system. So let's look at something like tube A generator. I can specify a particular number to generate, and then it will generate a barcode. You can also set up those barcode generators in the system in here and create them yourself. You can also set up your own container layouts. So if we look in here, so in here we should be able to, this is where we set up the prefix management. Here you can see a plate and rack layout wizard. So if I run that, give a plate layout, so let's say it's a 96 well, I don't know, demo plate layout. It could be a plate layout or a rack layout. As a description, we're going to say we're going to put lots of compound into it. We say the maximum volume and default volume for which controls how deep or shallow the wells are in the plate and the default unit. You then can specify either a default one or you can have a custom one. So I could leave that blank if I like, and then say, it's some strange plate format like 4x12, or I can say it's one of the standard ones. And then all we do now is basically lay out the plate. So I might want controls down this column, and then I say, okay, these are a high control, gives it a name, and then a fill direction. So we're going to fill them down. The block size is 1, or I can, and then a dilution factor, a number of replicates. We can replicate in doses, so that's replicate, or in series, where it's the series and then the series repeated. And then you can also have a repeat direction as well, and then add the block. So here you can see I've got them going down that way. I might want another one down this way. Same thing again, maybe the low control this time. Let's go down again. We'll use a block size of 8 this time. Add the block. So they're all the same now. If you've done something wrong, you can always remove the block and re-add it. So I'll add that one again. This time I'm going to make that 8. And it's high control. And then of course in the middle, I can define a dilution series, for example. So these are samples now. And we're going to go across the plate. The block size will be 10. And we're going to use a default dilution factor of 3.162 and no repeats. And I'm going to repeat in series this time. So add that block. And there we go. We've got a dilution series going across the plate. And if you hover over each one, it'll tell you what the dilution factor is. So in this way, you can define your own custom layouts in the system very easily and then set them up. So I won't keep that long. It's a demo. Okay.
Good. The next question, is there an API?
Yes, there's a full web service API to this. In the current version, it has a limited set of inventory features, but it covers most of the product. So if I look here and we look at this is the web service API that you can use in here. In the next version, we'll have the full support for all the inventory features as well. So that's being extended. So yes, there is an API.
Good. And then the next question, if you need multiple compounds but at different parameters, i.e. compound A is at 50 microliters at 100 millimolar, compound B 100 microliters at 100 millimolar, compound C at 100 microliters at 100 millimolar, can you request all these with the same submission, or does each compound need to be requested one at each time?
No, so when you go through the request procedure, so obviously the example I showed you was a highly standardized, higher throughput assay. So in that case, everything was at the same thing. But when I set up the service which allows me to request, you put in it the parameters that you want to vary. And then as you make the request, then you can specify the exact volume and concentration of every sample in the order if you want to do that. And it gives you a form to fill out that you can just fill out yourself. You can also map that form to a spreadsheet environment so you can fill it out there too, or you can automate it if there are rules to it as well. So yes, you can vary all of the order parameters actually per sample, and it's done through that mechanism I just showed you, which is setting up the order profile to take them from the parameters themselves. And then during the ordering process, you can fill out the parameters, the exact parameters you want for the order before you submit it. We're also working on some improvements to that as well, so the ordering forms and process will be more elaborate.
Okay. Next question, can I define the size of the plate I want to create? I think this goes to custom plate layout.
Yes, yeah, I think I just showed you that. So you saw the dimensions where I customized the dimensions.
Yeah. And how is security managed?
So the whole system has a security model. First, it starts with authentication. So you can authenticate against, you've got several options for authentication. You can authenticate against your LDAP system, so your Active Directory on your internal systems, or you can authenticate internally against the BioRails system. So again, if you're working with partners and CROs, it's highly likely they won't be in your LDAP system, so you can authenticate them internally as well. A user is then a member of a team, and then there is access control on what they're allowed to see and do. So that's managed through the team structure on the tree here. So if I fold this out, you can see I look at the DMPK CRO team. Let's look at that. And then there is something called an access control list on that. So we look at access control. That allows you to specify whether you're allowed to read, submit, update, or destroy data in that particular area. So in this way, you can keep CROs and partners away from data you don't want them to see, allow internal teams to see what they're allowed to see. And you can narrow that down to individual users or indeed teams as well. But obviously you have to be an administrator to do that. So I'm logged in as Barbara, so you can see it says admin rights are required to edit the access control list.
Thank you. Is there notification of completion of activities, for example, play completed by email?
Yeah, so I didn't show that because it's a little bit difficult to demo with emails flying around, but yes, system has an email notification center in it. If we go down here, you see manage notifications here. So an individual user can opt in to a number of different triggers for receiving emails. So you can say on what schedule you'd like to receive them, and then you can submit to a set of different notifications when certain things happen. So here, for example, I want to trigger an email that will tell me when my samples have been ordered. So I'd say as soon as possible, trigger when it's in the ordered state, for example. And we're working to increase these as well. You can also subscribe as well. So subscription says basically send me an email like on a daily or weekly basis to summarize the activity on a certain area. So it might be like how many requests have been made for a certain assay or how many plates have been generated by compound management, for example. So they're all triggered. And when the email arrives, it's got links in it to take you directly to the information required.
And then can the request be assigned by priority?
Yes, so there is a request. All of the requests come with a prioritization. If I look at, let me just, I won't show it, but yes, there's a priority on there as well. It goes high, medium, or low. And obviously, if everybody puts them as high, then there's not much point in having it. But also, you can filter by the project, the requester, and other information in there by using the column filters in there. You can also create your own custom filters and sorts, so you can sort it in the way you want to do it.
Great. And how are available amounts at the powder store updated in BioRails?
So what happens is, as we make an order from BioRails, that order is delegated through a web service layer to the remote system. And then the remote system processes the order and decrements its stock. When it fulfills the order by shipping the samples to BioRails again, it then updates BioRails via another web service, which updates our local copy of the stock. So that makes sure that the information at the BioRails end is synchronized. If we ordered, say, 5 milligrams from the powder store, and during weighing, one of the samples was dropped, so although we're getting 5, we actually took 10, then all that information is tracked as well, and then our local inventory levels are updated. So there's a supplied amount and a decremented amount, which is separate to make sure we synchronize the inventory. There's also a back-end procedure to allow you to run a synchronization process, which basically is a background job which will go through and ensure that both systems are synchronized and kept up to date.
And I think also on this point, can the processes be automated?
Yep, they can be automated via jobs that run in the background, or also they can be automated through the web service API as well. So we've got some of our customers are using robotic systems to actually automate the liquid dispensing and liquid handling operations, and they're pulling information from BioRails about the plates to create, and also updating BioRails with the information about the created plates so that the information is decremented.
Good. Thank you. Can work orders and cherry picks be sorted and configured for manual processing as opposed to an automated tube store?
Yep, so there is support in it for cherry picking. I didn't show you all of the wizards that are involved, but there are file-based mechanisms as well. So container from file and transfers that can be used to do more cherry picking operation. So that's pulling, you can pull tubes from standard racks, cherry pick them from the racks, and then prepare plates from those as well.
Good. Can it interpret an output file, i.e. log file, from an instrument to update the system of what it did, or does that need to be updated manually in the, bear with me, in the system as in the demo?
Yeah, so in the current version, there is a file-based mechanism for doing that. So you can automate the operation, collect up the sort of decrement operations that we need to do, and then you could just import them from a file that will update the system. We are extending the web services though to allow you to fully automate that whole procedure, so it's a lot less manual in the next version.
And to that, what's the expectation on the next release?
So we're working towards that. Will be out in I think towards the end of Q1 into Q2 next year.
Good. Well, that concludes the questions, and I think we're a little bit over, but I really want to thank everybody for their attendance and also participation and the number of questions. If there is anything that obviously we've missed, please feel free to let us know by email or call us. There is also a second presentation in about 50 minutes, so if you didn't get enough, there's another shot at it.